different techniques which are used in the advanced biology to study the cells, viruses, and the bacteria. In this segment, we will discuss an important technique by the name of Immunosorbent Electron Microscopy, ISEM. Immunosorbent Electron Microscopy. Now, immunosorbent electron microscopy is a diagnostic technique, and that is the diagnostic technique to use primarily in virology. You see, we have been previously discussing the cells, the whole cell, its behavior, the antigen concentration, and so on. But with the help of ISEM, we can diagnose, we can identify the different viruses which are present, or the virus-specific antibodies which are there. And how we can do this? Before we diagnose a virus, we get the virus-specific antibodies, and then we conjugate them with the gold particles. Take the virus, take its specific antibodies, and then conjugate it to the gold particle rather than to a conjugated enzyme or color-giving enzyme and things like this. And these virus antibodies are then used to visualize virus particles on electron microscope. You see, once we conjugate the antibodies on the gold particle, then we can visualize. Actually, we can see on a electron microscopy the virus particles which are there. And we conjugate those antibodies with the gold particles because the gold particle is electron dense. What we mean by electron dense is that where electron cannot pass, rather electron will give a black image, just like light. When it passes through, it cannot pass through opaque particle. So similarly, the gold is a electron dense compound, and it is seen as a dark shadow against the light background, because it doesn't let electron pass through it, and in the light background of the specimen field, the virus will appear as the dark. Now, technique is used in both transmission or scanning system, that we can use both in transmission electron or in a scanning electron microscope. That is, the both microscopes or both techniques can be used for this. Now, if gold labeled primary antibodies are not available, for example, if are not available for that virus or like that, then anti-immunoglobulin G gold conjugated antibodies that can be used to visualize the virus. That is what? Anti-IgG, anti-immunoglobulin G gold conjugated antibodies, they can also be used in this. They can be conjugated with the gold particles and visualized under the electron microscope. Now, these anti-IgG-gold conjugated antibodies, they can be used with the primary antibody in a double antibody system. So we can use a double antibody system, and these IgG gold conjugated particles, they can also be used with that primary antibody which is there and in that system. Now, another thing is that both monoclonal and polyclonal antibodies, they can be used by immunosorbent electron microscopy. This is the advantage of this technique, that both monoclonal and polyclonal antibodies, we can use them, or we can see them, or we can determine them by ISEM, immunosorbent electron microscopy. So this is a diagnostic technique which is used in the molecular biology.